Welcome to Under the Microscope. This series is brought to you by the Real Scientists Nano team. Our goal is to provide a platform where scientists can communicate their work and interact with the public. With that in mind, every week we introduce you to a scientist working in the field of materials and nanoscience.
Hello everyone. Today we have with us Craig Bunt, who is an associate professor at Lincoln University in New Zealand. Hi Craig, how are you doing?
Hello. Good, thank you. Hope you're well.
I'm well. I'm looking forward to speaking with you. Let's start with understanding your scientific journey so far. So how did you end up in your current research field?
If we go all the way back to when I was 15, I went to a careers night. There was a presentation from a pharmacist. So I was in middle high school. And I thought pharmacy sounded really interesting. So I had pharmacy in mind. I completed my high school with mostly science papers, and so went on to university for pre-pharmacy and completed a pharmacy degree. But partway through my pharmacy degree, I had a series of summer studentships, summer research positions that piqued my interest more in pharmaceutics than the pharmacy profession, the healthcare profession. So instead of becoming a registered pharmacist, I stayed on at university and went into a PhD and did my PhD in pharmaceutics, looking at microencapsulation for vaccines. New Zealand, a small country with not a particularly large pharmaceutical industry, certainly not a large pharmaceutical research industry. At that time, 4 million people. But the joke is 60 million sheep. It's actually less than that, but we have a very large veterinary pharmaceutical industry. So when I finished my PhD, I went and worked for a veterinary pharmaceutical company on controlled release inserts for estrus control, estrus synchrony. So timing when an animal can be inseminated or would breed, or for out of season breeding. So it's used as a farm management tool. And I fell on my feet there. It was a company that was reformulating its product for the US market. So I was on the team doing that. We patented what we were doing. So from a pure research PhD into industry, taking something to the US market, patenting. We were the first New Zealand company to achieve US FDA registration for a pharmaceutical, veterinary or human. This is 15 years ago now. And that was just an amazing 10 years of my life. And an amazing company director who just said to the scientists, you do science. You know, he followed the hard trained, capable people and let them do their job. Don't tell them how to do their job, and it was wonderful. The nature of business is that things change, so the research company became less active as its technologies were licensed off to large companies, Pfizer, Pharmacea, and the likes. So I moved on to academia, taught pharmaceutics at Auckland University, because up until that point, out of my PhD, I'd only ever worked for a small company, a team of 4 or 5 around, a few student interns and things like that, and I thought, let's get some experience in a large institute. And so New Zealand's large institutes are either retail, construction, or universities. And I was there for 2 years. That was amazing. I arrived in January and started teaching in February, the least favored course by pharmacy students, which was pharmaceutics, ironically. But most pharmacists aren't pharmaceutical scientists. So. But I brought in a lot of my industry knowledge and experience and hopefully some of the excitement. And so I turned the course around from the least preferred to one with no issues. Well, that's all you could hope for. But it was an amazing 2 years there. Then an opportunity came up to join a government research institute. And back doing some sort of research associated with what I'd done a bit of my PhD on, and microencapsulation of bacteria for vaccines. But the job with the Government Research Institute was taking bacteria and formulating them for green agri-chemicals. So bacteria to control plant pests, bacteria to control pest insects and things like that. So alternatives to agri-chemicals. I was there for 4 years leading a team to do that. And then another opportunity came up to go to academia, literally across the road from where I was to go to Lincoln University. It was, I remember at the time, $52 more a year. So I had a price, evidently, that would convince me to move, $1 a week. So I just remember it as quite ironic that I was going from one job to the other across the road, and I was gaining $1 a week. And I use that to illustrate that, you know, you shouldn't always be changing jobs just to chase the money. If the money is hard on what you're on and you can't live and it's a struggle, you're going to need to move. But sometimes job changes have nothing to do with money. They have a lot to do with where you need to be and want to be. And I'd been in government research, and I'd been in private industry, and I'd been in academia before. So I went back to academia, knowing of the three that that's the environment I like to be in most. Okay. So that was 9 years ago. And so here I am, having gone through all of these career changes with a background in biomedical pharmacy at an agricultural college. What am I doing? So I'm teaching physiology to animal scientists at the undergraduate level. Where am I going to take my research? And I was a little bit like a kid in a candy shop. There's lots of opportunities for formulation science. I'm ultimately a formulation scientist. You give me a piece of candy or a tablet, I'm looking at it and going, well, how was this constructed, and you know, what's it going to do once we take it? So whether I'm formulating a tablet or a fertilizer granule or hand sanitizer in our current environment, or a spray-on gel to control a pest on a plant, the formulation science is all the same, which is why my research now seems so varied, from 3D printing to nanofibers for ocular drug delivery for fertilizer, which does not sound all that exciting. But when you think of the need, yeah. So I actually, in some ways, dealing with the human body is so much easier. It goes in, we can model where it's going to come out, and things go along pretty much predictable pathways, because the human body is always trying to maintain homeostasis, and that we know how it should be, and when it's abnormal, what constitutes abnormal. I was doing a bit of field work a few months ago, and we're standing there in the middle of a drought, and my postdoc looks around and goes, but this could all be different next week. And I go, yes. And so what's happening this week? And I go, very little. The ground's so dry, nothing's happening. What if it rains and floods next week? I said, totally different. We'll have no idea what our material is going to do from 1 week to the next. Sure. Where if we put it in a human body, 37 degrees, pH 7.4, so enzymes and macrophages, yep.
Standard.
Standard. Yeah. So in agriculture, usually you have to do something 3 times, not for replication necessarily, but just to find 3 vastly different conditions to know really what the limits are. So yeah, it's my research field, my how I've got to where I am.
Okay, that's quite an interesting journey you've had. That's a really, really interesting start, and it's really interesting that you also worked at a private company, a government institution, and then you came back to academia because that's where you thought you belonged. Where and how does your current research fit in this big puzzle of materials or nanoscience?
So agricultural chemicals products have been around not for necessarily a long time, but they certainly burst on the scene more and more with the green revolution of the early 20th century. But a lot of the products you would see a farmer spread on their paddock now, give to their animals, were developed back in the 30s to the 50s, maybe 60s. And they're old technology. They're robust in that they can be transported and manufactured easily and put in the back of a spreader and put out there. But they have a lot of inefficiencies, a lot of dust, a high degree of so-called fail rate among. So if you have a whole beaker of granules, maybe 10-20% of them actually have all cracked and ground and don't actually perform as you would like them all to. But it doesn't really matter, because on the average in the whole beaker full of material, it'll still do the right job. Well, there's a lot of inefficiency in there, and there's a lot of waste, and that's not sustainable. So there's the need to take old technologies and make them better. And we can't make them better by still making them the same way out of the same materials, just putting another layer of something on. We need to do it a different way. We have the same issue in biomedical science as well. We have a molecule that needs to be in a certain part of the body. There's no point delivering it to other parts, or there is a molecule that's difficult to get to the right part of the body. So we've got an efficiency delivery challenge there as well. And so how we package, whether it's a bioactive drug or we package a fertilizer molecule so that we can administer it and deliver it, requires the field of material science, formulation science. So taking a pile of active and transforming that into something that can be used and do its task. So it's the chemical engineering of that and the material science of being able to select new ways to do that, because the old ways, a lot of them are exhausted. What I find really interesting is a lot of the bioactive molecules, there's old ones that we know really well, what they can do, how they do it, where they go. But if we can use them better, we actually probably have something far better than a whole new molecule. And what I'm saying there is, if we take a whole new molecule, we don't know what it does in the environment, what it does in the body. So even if we deliver it wonderfully, we may find other things are a problem. If we take an old molecule that we know it has limits because it washes away too fast or doesn't dissolve fast enough or whatever reason, but then make that hang around long or go quicker, then we still know what the downstream consequences are, or we can manage them and accept them. So material science, formulation science also enables you to develop or extend the capabilities of a molecule, extend its product portfolio. The story I often remember is Pfizer's very first blockbuster wasn't a new drug. It was taking an old, well-established drug that you used to have to give 4 times a day and putting that in a tablet where you only had to give it once a day. And that's convenient, but also at the same time, they found that it had other health benefits. And they already knew this molecule, how any problems or how wonderful it worked, but they actually made it work better, not by creating a new molecule, but by better formulation. So
modifying the known molecule and making it better, and instead of starting from square one, you start with the existing molecule and then make it better and fix the problems that you know, and not create new problems by going down a different path altogether with a completely new molecule. That makes sense. And it's also cool that the material sciences, the material is what that's the commonality, be it agriculture, be it biomedical or pharmaceutical sciences. That's also really, I think that's a very good description of where your research falls in this big puzzle of materials or nanoscience. So it sounds to me like you do a lot of interesting experiments and field works. And if you have to pick one research project, a quirky one or a fun one, or the one that you're most proud of. I know it's very difficult because you do so many interesting research projects. Could you pick one and explain it to us in simple words in the section we call In Other Words?
So this is going to be controversial to many people. but I'd like to give a bit of background first. So in New Zealand, we were colonized by Europe 200 years ago. Before that, we had the indigenous Polynesian people, the Maori, had been here for 1700 years, maybe a little longer. They brought with them the Polynesian rat and the dog. And that was about the only animals they brought with them, and some plants from the Pacific Islands. So by the time the Europeans came here, there's this unique flora and fauna in New Zealand of plants and animals, birds mostly. The only mammals prior to European settlement were humans, dogs, the introduced Polynesian rat, and 2 small bats. That was it. No land mammals.
Wow.
No foxes, no bison, no wildebeest, no giraffes, no Norwegian rats, no ship rats, no mice, no mammals. The Europeans bring in a whole series of different animals for different reasons. But they brought in the rabbit to make New Zealand look like England and provide a source of meat. They brought in the sheep, and then they brought in game animals, deer and chamois and tar and pigs, and the possum. North America, there's a mammal called the opossum, but in Australia there's a marsupial called the possum, a furry animal. Looks a bit like a vicious, mad squirrel with sharp teeth and claws. Can look quite cute, actually. Usually in Australia, where it's native, any photographs show it looking very cute and friendly. In New Zealand, we don't tend to like those photographs because the millions of them in our forests decimate the bird populations, the trees, and so we have a major pest problem in New Zealand of introduced pests. The mustelids, the stoats, the ferrets, rats, mice, the possum. We even have wallaby in New Zealand. Now, Australia has wallaby and kangaroos. We even have wild wallabies in New Zealand. So all these pest problems. And so New Zealand has embraced the to be predator-free by 2050. To remove these predators we've introduced and are damaging our ecosystems by 2050. And that will be done through a whole range of activities, by fencing off large areas of land and taking the pests out, eradicating them, by hunting, by trapping, and by poisoning, letting native species regroup, repopulate, and then move out across the country and remove these pests. And so New Zealand uses a controversial mammal control toxin called 1080. We're one of the world's largest users of 1080 for controlling mammalian pests. And there is always a need for alternatives to things like 1080. 1080 has a lot of really good characteristics in terms of speed of kill. That's controversial, but in terms of environmental residue, it's non-existent. It's quite more specific towards mammals than birds. The animals we're trying to protect are birds, and so we have this unique situation in New Zealand. We don't really have native land mammals to protect from 1080. And the few examples we do have, the bat, we can keep the 1080 away from them quite easily by making a bait that they wouldn't go near. So there's also our use of toxins, but there's also exploring new toxins, more environmentally friendly, more humane, more sustainable. And so some years ago, I was having a conversation with a company looking at trying to develop a new toxin for pig and possum control. And they were looking at a compound called sodium nitrate, and that's the preservative you'll find in processed meat sometimes. A small amount is a preservative, a large amount is a toxin. Now that's the case with any chemical, salt, water, sugar. Small amount versus large amount. What's the definition of a toxin? Enough of it will kill you. So sodium nitrate, it kills by basically sending the body to sleep. It doesn't hang around in the environment. Nitrates are there anyway. Nitrates are produced by plants. Has an antidote, though, if there's accidental poisoning. There's an antidote available for that. But it's incredibly bitter. So how do you get a pest animal, to eat it? And so the company was looking at coating the granules of this poison with a material to taste mask, and they weren't having very much success. They knew they were getting some benefit, but not as much as they needed. Animals could still taste this and then move away from the bait, the toxin. So I just talking with them, and they said, what could be happening? I said, well, they described their coating process, and I said, well, maybe you're getting cracks in the coating. Oh, how would we know that? I said, we'll look at it under an SEM. Okay, and then they say, well, if we find cracks, how would we fix it? And I said, well, maybe you need a plasticizer, something to help the coating material flow over the granule and form a better, more intact coating, one that sort of stretches over rather than just lies on there, so that if it can stretch and flow over, it won't have cracks. Okay, what sort of plasticizers would be? And I just, off the top of my head, listed off 3 or 4. Or how much would we put in? So I just gave them a ballpark figure of 1% by weight. Okay, and the company director's writing down these details, and I just thought it was a conversation. Once I start talking, getting me to shut up is impossible. And then 2 weeks later, the director contacted me and says, oh, we have some samples now. Can you test them? And I'm, what? And, oh, we've made these samples with this formulation you described. And I'm thinking, oh, gosh, I hope I'm right. So,
were you?
Yes. One of the plasticizers and the concentration produced a coating without cracks.
Wonderful.
And so, yeah. And so that formulation then went into registration trials, and so it's now used in Australia to control feral pigs. It's registered in New Zealand but not used.
Okay.
Now this is interesting. The reason it's not used is not because it's unsuitable or anything like that, is that we have existing control programs. We understand the science of really well how to use it, how to use them. And so if you introduce a new compound, even though it may in trials work effectively, you don't know exactly how you're going to use it out in the wild, convincing these wild populations of animals to engage with it. So we're still sticking with the science of the known products, but it's registered for essentially in an emergency. New Zealand's foot and mouth free. We don't have that here. And so if it was to come into the country, how would we control it? Now we control it in farms where there's sheep and cattle, but the worry would be feral populations of pigs. How would we control it there? We can't hunt them all. New Zealand's too big with rugged mountains and deep bush. So the product is registered in New Zealand in the anticipation that, well, what if we needed to control the feral population really quickly? It'd be hugely expensive, but we could use it that way. And it's also going through US registration as well. So it's being trialed by the USDA and various sites in the US. And so it's not actually outstanding science, but I like that project because we had the right people talking at the right time for the right situation, this need that needs to be met, and a simple solution. And then seeing where that solution ends up in terms of products and trials and providing more options. So that's the one I'm most proud of.
I can understand why you're most proud of it, because also the turnaround time of 2 weeks, almost 2 weeks, at least for the first trial, that's also like, wow, that's incredible. I can completely understand why you picked this and gave a disclaimer at the beginning that the situation is different in New Zealand. It might not be the same in other countries. So the giving context is often very important. This is very, very good. Craig, let's move a bit to your other parts of your work, which is teaching. Do you teach courses? And if you do, which one would you like to mention? Which ones would you like to mention?
Yep. So I teach some undergraduate classes. I'm the examiner for one of the courses. I'm the lead teacher for a first year. foundation paper in animal science, in agricultural science, and the paper I teach is animal science. It's essentially mammalian physiology. And then I teach a little bit into some third year and fourth year courses around lactation and endocrinology and rumen, cow stomach physiology. I'd like to talk mostly about the animal science first year teaching. And I find that most exciting and most frustrating.
Why?
So I had taught previously at Auckland University in the School of Pharmacy, and that was my first university teaching engagement, teaching pharmacy students who were in a restricted entry course. You could only do pharmacy with having gone through a process of applying your grades and an interview to even get into the first year. So 100 places per year, 4-year program. I was mostly teaching third and fourth year pharmacy students, professionals, already professional people who had to highly achieve to get to the course. So, I would get back to my office after a lecture and there'd already be a queue of more questions on my lecture and stuff like that. And then I went to an agricultural college where it's a general entry course, first year students, and I describe the class as divided into 3. There's a group of students who just need me to point them in the direction, and they will go and they will achieve and apply themselves and study and do far better than I ever did as an undergraduate. Then there's a group where we'll get there together. We'll work on it. We'll keep talking about it. We'll learn together and pass the course.
Right.
Then there's a group who I say are just there for the lifestyle. They're either there to experience university, or they're there because their family expects them to be there, but they're not necessarily convinced. They're not actually quite sure if they will finish university, if it's the right course for them. So I wasn't prepared for a general entry course where there's going to be students who fail, and there are going to be students who are worried about failing and want to pass, and you can work with them. But then there's a group who just don't care.
Right.
and so I know as teachers we're often told you have to find a way to get through to them, and what you have to accept as a teacher, sometimes you can't get through to them.
True.
And getting through to them might not be the answer, because helping them achieve in a course ultimately that they may never stick with. So sometimes it's helping them realize this is not the course for them, that if they're not applying themselves and they're not trying to pass, then they'd be better off using that time applying it to a different course or a different career or whatever. But also letting them know that, yeah, they're going to fail the course. That's not a reflection on them.
Right. It's not. It's just sometimes it just doesn't fit. It doesn't click. And then one should know where to draw the line of pushing and helping the students. Yeah, I think that's a very important point that you raised. That's very, very important. I can understand why you wanted to talk about this entry-level undergraduate mammalian physiology rather than teaching the professionals, because I'm sure that must have been super frustrating.
Yeah, and senior year students, they've already got the lay of the land. They know how to be a university student, what's expected of them. It's the first year students who have come from high school and been taught in a certain way for 4 or 5 years, and now go to university where a lot of it is self-directed. And it's yeah. So but at the same time, that's incredibly exciting because you have the students who just totally engage from day one. And I have a big class, so enrollment can be anywhere between 150 and 250 students, depending on the year. So in a small class, if there was 90, 20 or 30, it might be 1 or 2 who are really, really struggling, or it might not even bother to turn up. So you don't know about them. But in a class of 150, the ones who are struggling or not applying themselves, you know who they are straight away. you can spot them. Just
Right.
plot out the
Yeah, statistically it's more, right? 1 out of 20, multiply that by 5,
yeah.
it's suddenly becoming 5 out of 100. Yeah, yeah.
And anything you try to address that will find some success in a big class. There'll be a group of them where you try something new and they will click. Where if you have a small class, you try something new, but it still fails 100%. But that's just because there was only, say, 3 students. And
Right.
where you try the same approach in a large class, where there might be 30 students like that, and 10 click, and you've helped that 10. So it's science. The bigger the numbers, the replication.
Exactly. Yeah, and that's all part of the process. Absolutely.
Mm.
Craig, I hope your research experience so far, let's move a bit to the research experience now, has been wonderful so far and will continue to do so in the future as well. However, if you had 3 wishes to improve your research experience, what would you ask for? And I'm not promising anything here.
Yeah, funding. Gosh, it's difficult to apply for now. You know, it used to be you wrote a letter explaining what you were going to do, and that was it. And now funding applications run to 20, 30, 100, 200 pages, and there's sections on business and impact and risk and alternate. And so not only are we scientists, we have to do a bit of economic modeling, we have to do a bit of marketing, future projection, not only prove we've already done it and can do it, but that if it doesn't work, we have a backup plan that'll be just as successful. So is there a way to actually remove the bureaucracy between the applicant and the funders? Because there's this whole body of bureaucracy now that never used to exist. Do they really add value? Are they needed? I worked for a government institute that vast majority of its funding was applied for externally, secured externally, but from the government. Our funding model is, even though you have government institutes, they have to apply for public funding.
Right.
And that was this process of having to apply and the bureaucracy and the big applications was frustrating to the scientists. And then this government institute got a big bulk of what was called core funding, where it was no longer publicly contestable. The institute was able to decide how to spend it for research.
Okay.
But the scientists still had to go through the same sort of process that the external one was. Basically, the government institute took the external process and changed the letterhead. So it made no difference to the scientists having to go through all this bureaucracy. So can there please be a way to, if not remove the bureaucracy, protect the scientists from it? That would be good. I'd like better networking between scientists, amongst scientists. It's amazing the number of times I've been away to a conference, you're having a conversation with somebody, you realize you know somebody in common, but then one of them, somebody you point out, oh, you should be working with so-and-so on such and such, and you go, are they doing that? And they go, yeah, didn't you know? And I go, no, they're down the corridor from me. Why didn't I know this? You know, so scientists are really bad at telling others what they're doing or what they can do, and we stand behind our capability, we stand behind our data, but we need to somehow step out in front of it more often. That's a double-edged sword, though, because we could be over-promising, over-delivering, and not aware of our limitations. So, you know, but and now we have all the platforms, the LinkedIn, and using Twitter professionally or socially, and every conference seems to have a different app to then work out what sessions you're going to go to, and you know how many different places are there now to register a profile as a scientist. So you know, yet it's still difficult to find who's doing what and who's capable of what. So better networking, because networking still takes a lot of effort and a long time. The process hasn't been streamlined. We just have different ways of recording business cards. And then the publishing process. Oh, please. It's so frustrating that you have a journal requires you to format it to such and such before you instantly rejected by the editor, and it's just like, well, if you told me that 3 weeks ago, I wouldn't have. And then you got to reformat for the next journal. Is that really needed? And it's not, because reviewers aren't looking necessarily, is it formatted according to the journal's guidelines? We're often asked that, but really it's irrelevant. And we should be able to submit in any format we want. And if we do a bad job, it should be on us. If we've submitted something that can't be read, its structure is a nightmare, the referencing is, and it's instantly rejected because of that. Well, that's fine. I should put the effort into presenting it in a way that is readable, but not to fit the journal requirements. Yes, it's rejected. Then I can just submit to the next one. I could submit to the next one and the next one. So I know amongst scientists that's right under the frustration of reviewer 2. It's having to reformat for another journal. I'd wish the need to format to go away, and I'm tied up with that. I'm not entirely convinced the pay thousands of dollars for open access is a better model than the paywall, because you've just moved the wall and created a barrier for many scientists against open access publishing. So the institutes who could pay to get behind the paywalls, the institutes who can pay for open access, still get their information out there one way or the other. The institutes who couldn't pay to get behind the paywall can no more pay for open access. So their results, their work is still hidden somewhere. So I'm not convinced that the open access pay model at the moment is the right one.
So these are your 3.
So I've a wish there, then not for a solution, but I've a wish there that a bit more effort was being put in, that the pay for open access is going to free up science. It's not. It's still going to lock away a lot of science, particularly. You may have a project with a student who's got some great work you want to publish, but it has to go behind a paywall, because where are you going to find the extra few thousand for open access? Not every researcher has access to that money, nor should they need to. We have to add another budget line in our funding application, which just means something else has to be reduced.
In the funding proposal. Yeah, 102 pages instead of 100 pages now for the funding. Yeah, absolutely. Yeah, all 3 of your wishes are absolutely fair, and I wish I could change that. I wish I could just do it with a snap of my fingers. But I would like to believe that we as a community, a scientific community, we're working towards achieving that. And going towards more fairer, less amount of bureaucracy and different structures of funding, basically making really opening the access to all kinds of research work, not just the ones where the institutions have the money to pay for it. I completely, it resonates, absolutely. I'm sure it will resonate with a lot of researchers as well. So very, very important points that you brought up, also with the networking, very, very important points. Speaking of future, what are you most looking forward to in the next 3 months?
This COVID epidemic to be well under control, if not eradicated. That's in everybody's mind at the moment. But some good things will come out of this. Maybe not as big as we would hope in terms of social changes. And we're human, we'll fall back to our old habits. So I think the air pollution will come back, the water pollution will come back, the noise in the cities, that will come back. But it has been a crash course in truly working from home, not just going home because there's some quiet to do some marketing. No, truly working from home for many of us who occasionally would say, I'm working from home, but you weren't. And we're now experiencing what it's like for those who before this were successfully working from home, who had done that. And so I'm hoping that coming out of this, that the new skills I've acquired and the new tools for working from home, I can maintain those. And so I'm finding now I have a far better routine to my day than I do at my workplace.
Oh, interesting.
Nobody's popping into my office with a quick question. There's a downside to that. The email spam has dropped off amazingly. I think some internet cafes in some part of the world are obviously closed, and the spammers aren't as active as they have been. So what gets through the spam filters is nowhere near as much as I've had in the past. So emails have quietened down.
That's good.
But using Zoom and MS Teams and software and coordinate and sharing folders and files far more efficiently than in the past, and just the learning curve from where I was 2 weeks ago to where I am now. We so many of us know how to change the backgrounds and things like that, but just being able to set up a meeting and knowing what's the advantages of Zoom versus Skype for Business versus MS Teams, difference between just shared folders or SharePoint sites and things like that, and finding that's actually improving how I certainly work with my team.
Okay, interesting.
Yeah.
Interesting, very interesting.
So I want to maintain a lot of that. Besides the decor and the heating and the IT support in my home office is far better than at work. So I have to say that because I'm the IT support here. So.
Oh, okay. No bias, no bias there.
No bias.
Absolutely. Okay, that's wonderful. And before we let you go, Craig, what we want to understand from you is what are the big challenges faced by the field of materials or nanoscience, or the big questions that the scientific community is working towards solving, other than the vaccine for COVID-19?
Yes. One of the biggest challenges, I think, outside of the laboratory for nanomaterials is their true environmental fate. It's not just that is this chemical used to make a nanofiber present in the environment. It's where is it actually going? It might be stuck on a granule, but then gets engulfed by some microbe and partially enzymatically degraded, and then bits of it go elsewhere. And we can track the chemicals, but we can't necessarily track what they've gone through.
Right.
And we know that if we look at a paddock, most of the biomass is under the grass. There might even be cows there, but under the grass, there's the worms and the other invertebrates and microbes, and the diversity there is just like staggering. If you physically put it on a scale, what's under the ground is enormous compared to what's above the ground. And we know chemically what might go in, and we might know the chemical fate of materials, but we don't know what organisms they're going through. We might be able to get an idea that some genes there are responding in a certain way that maybe have something to do with what we put on, maybe not. Maybe because the wind changed direction and dried the paddock out, So that's a real difficult challenge to understand nanomaterials, and ultimately the fate of any material we as humans produce. We could start off with the biggest thing, but if it erodes, eventually it's probably going to turn into some sort of nano presence. And then there is still the struggle to get research translated to industry. From the research lab and industry, you've got two bodies who work at different speeds with different priorities. There's always the idea of, well, if you have a student based in industry for part of their studies, or scientists seconded to cost them, that's going to help. I think that helps identify the differences and the struggles to communicate, but I don't necessarily feel confident that it improves the ability to translate research into industry. And there are so many different models in the world that you could try this and try that. It still comes down to a lot of luck, right people at the right place at the right time, the right need. A lot of the time you see things literally reinvented because others weren't aware that this bit of research was done prior to things being scanned and put into Google. It's still sitting in a library somewhere. So there's still that struggle as well, and that's an inefficiency in the way we as humans spend our money to do research and then make the research of use. No research is wasted. Every bit of research has a use, even if it is just to train the researcher to be a better researcher. And that applies humanities, art, science, engineering, maths, technology, the whole lot. No research is wasted. But research is lost and forgotten and not realized what it can be used for or what it means and things like that. So translating it not just to industry, but translating to it into a way society can use it and access it and gain from it. Then we have to guard against the misuse of science, the misappropriation of science, and as we get science into industry and products better, it'll get misused even more. So
Absolutely,
yeah.
That's a lot of challenges. So there's still a lot of work to do for the researchers and the scientists. A lot of challenges and questions to be answered.
Well, isn't it frustrating when some politicians and bureaucrats sometimes say, "Oh, we don't need to fund this field of research anymore. It's already all been done." No, it's never done.
Yeah. I share that frustration, absolutely. Let's not go there. But I hope we are working towards solving these problems and keeping sane, keeping our moral compass in the right direction when translating the research from the lab to the market, to the lab bench to the market shelf. I hope we are working towards it. On that positive note, thank you, Craig, for speaking with us. This has been wonderful, and we are excited to have you on Real Scientist Nano.
Thank you. Thank you very much. I'm really looking forward to it. Wonderful.
Thank you for listening. To know more about us, please visit our website, realscientistsnano.org, and follow us on Twitter at realsci_nano.